Good afternoon. I want first to thank the organizer for the invitation and giving me the opportunity to share with you our work. So we changed a little bit the title. It's New Advances in Pathophysiology and Therapeutics, because the advances in therapeutics in Prader-Willi syndrome really derived from the advances in pathophysiology. This is my conflicts of interest. So Prader-Willi syndrome has changed from obesity, very severe obesity, as the leader of the syndromic obesity, to now a disease that is diagnosed very early in life. In France, we made a survey a few years ago showing that the genetic diagnosis is made at 18 days of life. So I mean, it's not a syndromic obesity that we diagnose, but a child with a neurodevelopmental trouble. So Prader-Willi syndrome is a rare genetic developmental disease due to the lack of expression of some paternal alleles on the chromosomal region 15q11q13. It was the first imprinting disorder described in humans. And I will want to show you that we have now strong evidence supporting that the hypothalamic dysfunction drives the whole phenotype. So from a genetic point of view, I'm not going to go into detail. I'm a pediatrician. I know genetics, but I'm not an expert. So the PWS region is a very complicated one, at least 20 genes in the locus. And some of the genes of the region are imprinted, maternally imprinted. That means that they are only expressed from the paternal chromosome. And there are many different genes. I don't know if I can use that. Perhaps here, right. So these genes here in blue are expressed in the brain, and these genes here, the small nucleolar RNAs, are also expressed in brain, but not only in brain. And we will go back in a moment. And these are the other genes in the same region. And the Angelman disease is due to the deletion or mutation of the UB3A. So this is a very complex disorder from a genetic point of view, and I will show you that it's also very complex from a clinical point of view. So we have now, it has changed from many years now. When we diagnose a child, he can have a deletion from the paternal chromosome or a disomy, a maternal disomy. It's not duplication, as you just recently heard about, it's a maternal disomy. So the ratio has been changing due to the maternal age. For many years we have think about, do we have a minimal critical region that drives the complete phenotype? It was difficult, so analyzing the chromosomal translocation, we are able to identify a very small region in this chromosome, including the SNORD11 cluster, the SNORD109A, and the two exons of the IPW gene. And interestingly, when this deletion is deleted in mice, it completely recapitulates the PWS phenotype in mice, which is not exactly the same than in human. And fortunately, we were able, some years ago, to find a person, a young woman, who has the same deletion that the minimal critical region. It is non-genomic imprinted because the profile was not abnormal, but the deletion came from the paternal chromosome, so from the imprinted gene. And this young woman has a complete PWS phenotype, which is not light, as we thought before, so exactly the phenotype of Prader-Willi syndrome. That raises the question that probably this gene, this SNORD116 locus, drive the entire phenotype of Prader-Willi syndrome. It has to be confirmed yet, but we have now many evidences showing that. So we have been we have been lucky to work with the team in Florida, in Gainesville, and describe the natural history of the disease from birth and from before birth also to adults. And what John Miller described, different phases ranging from hypotonia with difficulty of feeding, just postnatally, to phase 4, appetite, phase 3, hyperphagic, very strong appetite, no satiety. And what we proposed some years after this publication in our team is that we can identify two main phases. One characterized by hyperphagia and behavior troubles, and the first one just after birth and probably before birth, which is characterized by anorexia and poor interactions. It was the first time that we used the word anorexia, and I'm going to show you what are the arguments that we have to name these as anorexia. And in between, there is a developmental switch that we are working on to unravel the causes and the mechanism. So what we can say is that verbal and social skills deficits observed in Prader-Willi syndrome in all the patients drive life-threatening and poor socialization skills. The hypothalamus probably explains all the symptoms. The endocrine and metabolic troubles on the left, feeding behavior and appetite regulation troubles, the general behavior troubles, I will focus after this slide, and dysautonomia. It's very important to focus on dysautonomia because it is not often described, and I really do think that it's not only in Prader-Willi syndrome, but also in other neurodevelopmental diseases that we have dysautonomia, which is not a diagnosis. So you can understand, you know, with all these symptoms that these patients deserve the comprehensive care, which is not always the case, but in our team for more than 20 years now, we are trying to develop this comprehensive care. So first, endocrine signs, endocrine disorders that we have to care. Then the behavior, which is very important to address, of course. We cannot care of this patient without a psychiatrist and pedopsychiatrist. Also, we have to care of dysautonomia, as I said, and many comorbidities that arise throughout ages. They are not related to obesity. That was thought before, but it's not the case. We can have many comorbidities without obesity, like, for instance, diabetes. It's not always linked to obesity in these patients. So very severe comorbidities, epilepsy, scoliosis, ophthalmology issues, gastrointestinal problems that really are very severe and unknown and skin-picking. And our view is that the behavior troubles are completely integrated with the feeding behavior troubles. I'm not going into details because lack of time, but as you can see here, these patients display personality traits and affective disorders. We talked about already this morning in the 22q11 deletions, with personality traits and affective disorders, cognitive deficits, which are not very intensive. It's mild or moderate intellectual deficiency in these patients. They also have poor social skills. And their feeding behavior has been really well characterized, thanks to a very good persons who work on it. And it's more an obsession with food and an addiction with food than a constant hunger. It has been really and smartly well demonstrated. They have a behavior of craving for food and storaging, not only food, but everything. They can take and storage everything. They have sugar preferences. They have a prolonged duration of meal if you not help them to make a fixed duration of meal. And they display deficit of satiety and hyperphagia. So how can we address all these symptoms? Regarding the feeding phenotype, we really worked to demonstrate that Prader-Willi syndrome is a model for understanding the gradient system, which is very complicated to understand. So I'm not going to explain everything in the gradient system, just to tell you that from the beginning, from 2002, it has been shown that the circulating gradient level is elevated in Prader-Willi patients. In pink, in yellow here, it has been shown that these elevated ghrelin levels are due to elevated increased acylate ghrelin level. These peptides exist and circulate into two forms. One acylated, which is the orexigenic hormone, the hungry hormone has been called when it was first described, and it's also circulated in an unacetylated form, which probably has the opposite effect. I will discuss about that further. It is still debated if these two forms of ghrelin really exist in vivo, or it is something that arises from the instability of acylated ghrelin. So I'm not going into details on that, but you will see the result that we have. And really, very early after this discovery, we were able to show that high ghrelin levels are present throughout life in this patient, even though when these patients are not hyperphagic, on the opposite, even in infants who are not hyperphagic but are anorexic, as I told previously. And what we did first with the Netherlands group, we demonstrate that measuring acylate ghrelin separately from unacylate ghrelin, we confirm that ghrelin levels are elevated in Prader-Willi syndrome. It is in blue here. But compared to controls, who are in green here, there is a relative deficit of desacylate ghrelin in Prader-Willi syndrome. This relative deficit is also observed in obese people without Prader-Willi syndrome here, but in the obese people, we don't observe high acylate ghrelin, but we observe a decrease of acylate ghrelin, but also a relative insufficiency of unacylate ghrelin. And this couple of hormones have to work together. And what is also really interesting is that we were able to show that the increased ratio from acylate ghrelin to unacylate ghrelin was observed when hyperphagia starts in these patients, so starting at phase 2A, and it is not documented here. What we did, because I am a pediatrician, we measure this ghrelin level from birth to 4 years old, and we're surprised to observe that at this time there is no differences comparing to age-matched control of ghrelin levels. They are the same acylate ghrelin levels. They are still total ghrelin level, but this elevation is due to the high unacylate ghrelin level. So at this time, it is the opposite. Ghrelin levels, total ghrelin levels are elevated, but it is due to unacylate ghrelin level excess. And we propose that this excess drive anorexia at these phases, at this age. Interestingly, regarding why do these people have high ghrelin level, the gene of ghrelin is not inside the chromosomal region. But interestingly, there is a mouse model of Prader-Willi syndrome obtained by a mutation of one of the genes that I showed you previously, the MAGEL2 gene, that impairs the development of hypothalamic anorexigenic circuits, but also display high ghrelin level, which is really interesting for us to see. And, and we worked with this team in France and in the US regarding this mice model. Interestingly, recently in 2013, this gene has also been shown to be mutated in some people with ASD. And interestingly, those patients who have ASD and this mutation display the neonatal feature of Prader-Willi syndrome. So this disease is called now Schaff-Young syndrome, and it's, for us, a PWS-like neonates feature. So, as I told you, the gradient is elevated postnatally and in adult of a mouse with MAGEL2 mutation here and here. Another explanation of an abnormal ghrelin pattern in this patient is the observation that we have been able to show with the team of Rudolf Lebel in New York and Columbia University, is that in iPS cell-derived neuron from the patients, we show the decrease of hypothalamic transcription factor NHLH2, which is interesting to see that this factor is related to the energy availability in the body. It's decrease in fasting, and the opposite increase with food intake. So these genes drive the expression of the PSK1 gene, so the gene who encodes the pro-converters C1 protease. This protease maturates all these peptide here into the mature peptide. I have to say here that ghrelin is not known to be synthesized by the hypothalamic neuron. It has been found, the pattern, the profile of expression of the ghrelin gene exists in the brain, but we are not sure yet that ghrelin is synthesized in the hypothalamic. And why when I tell you everything on ghrelin in these people, it's because for the first time we are able to demonstrate that modifying by a drug the ghrelin system may modify the feeding behavior of this patient. We use a synthetic product in a multicentric European double-blind phase 2 study. We administered to these patients this AZP-531, which is now called levolotide. It's a 8 amino acid small cyclic peptide, which is an analog of, I'm sorry, of the mistake of unacylate ghrelin. Sorry for that. So, and this peptide mimics all the effect of unacylate ghrelin. And particularly, it prevents the food intake after administration of acylate ghrelin. The mechanism of action of this drug is not, and of unacylate ghrelin is not completely understood right now. So after 14 days, it was the longest duration that we were able to make because it's a new drug. We were able to demonstrate that the hyperphagia questionnaire provided by Dykens was significantly improved in those patients who received in a blinded manner the drug. And particularly what was important is that the severe criteria of this questionnaire were particularly improved. And if you are interested in this kind of questionnaire, so it's many questions, it's 11 questions. Now we have modified it for 9 questions, and it's easy to see that it's really capture the addiction for food. Patients are also less hungry after having breakfast in the treated group and not in the placebo group with the visual scale that we made. And now, after having talked about ghrelin, I want to introduce the oxytocin, which is, I will show you, very related to ghrelin. It was really interesting to see that the oxytocin, which is a neuropeptide and an hormone also, secreted by the hypothalamic, also in the paraventricular nucleus and in the supraoptic nucleus, through different neurosecretion types, axonal and dendritic secretion. And from many years now, from 1995, it has been shown that people with Prader-Willi syndrome display decreased number and volume of oxytocin neuron in the paraventricular nucleus. And what was interesting is thereafter in 2000 and after in 2010, 2 mice models reproduce exactly the abnormalities of oxytocin neuron in the hypothalamus. And these 2 models are PWS mice models. And it is not the case with vasopressin, which is very close to the oxytocin, close by the amino acids, only 2 differences, and close with regarding the receptors and the neurons who secreted these 2 neuropeptides. In patients with Prader-Willi syndrome, there are not a decrease of oxytocin levels, but an increase of oxytocin levels, which we don't have the explanation for that. So since this time, since 2005, I must say, so we had many discussions on the Prader-Willi community and tried to start oxytocin clinical trials. We did the first one and published the first one in adults in 2011. And as you can see, there are many trials ongoing or finished. I will present you the data in infants because we are the only team working on that and obtain very promising results. But we have now many studies in children and adolescents. So why do we want to treat early these infants with Prader-Willi syndrome? First, because we can do that, because we diagnose the disease during the first months of life, and because we had very good data, preclinical data in the same model, major 2 cow mice. Françoise Muscatelli demonstrated that these mice, as pup, have a very early lethality and a very important lethality, because 50% of them died in the first hours of life. She demonstrated that it was because they have a sucking deficit. These pups have no feeding. And he can rescue completely the lethality after a single injection of oxytocin during a very short window of time before the first 5 hours of life. And when she followed these animals throughout their life, we published together that we demonstrated that administration early oxytocin in a short course completely rescue not only the feeding at birth, but also the memory, learning, and socialization in only male mice. So it was very interesting. It was our idea that oxytocin can drive postnatal effect and late effects or long-term effects. It depends the way that you explain it. And we published in 2017 the results of our second study in infants with Prader-Willi syndrome. We demonstrate in 18 children coming from all over the country to our reference center in Toulouse, less than 6 months, that in these babies shown here, we demonstrate that oxytocin administration intranasally improves feeding, improves sucking and swallowing skills, and also modified the acetylcholine levels. So it increases appetite, and it also increases the oral motor feeding skills. And we also demonstrate with our friends and colleagues here, with Antoine Gadonnet, David Cohen, and Sylvie Nivelong, that oxytocin improves social retreat and mother-infant interactions by using, in a blinded analysis and in video, the alarm distress baby scale. We did not normalize this scale in every baby, but we have 81% normal score after oxytocin, while having only 20% with normal score before. And using the coding interacting behavior scales, we demonstrated a significant improvement in child state, social engagement of the child, parental sensitivity, and dyadic reciprocity. And interestingly, when we were able to measure with the functional MRI studies, we demonstrated a significant increase in neuronal connectivity of the right orbitofrontal cortex, which is a brain region of importance, particularly in feeding and social skills. It has just been shown in 2019 that in this region, there are neurons involved in feeding and neurons involved in social skills and the interaction between both of these neurons. We also showed that the changes in the score of neuronal connectivity correlates with the changes in the normal score, so the sucking and swallowing skills, and the CIB teams, particularly those addressing the child's social engagement and social states. So it was very interesting to show for the first time in this population that oxytocin may have some very promising effects. If we have time, I can just show you this child, this infant. He's a boy of 1 month and a half. He was born in Toulouse, so that's the reason that we included him very early after birth in our study. He has like 80% of the babies a nasogastric tube feeding. He has really severe hypotonia. Very difficulty for him, very difficult to look at his mom, but he can try. And now the physician tried to stimulate him because normally he has to be fed, but he didn't demonstrate any hunger sign. He didn't have any reflex when we tried to obtain that. And then when we proposed the bottle, it is very difficult to obtain sucking movements and swallowing movements, even though when we put the bottle into the mouth. So it's a classical features presentation of a baby with a Prader-Willi syndrome. And I will show you not the results on the second swallowing because of lack of time, but the results on the social skills, because I thought that you are probably more interested in that. So this baby was in the third step of the study. So he received for 7 days 4 units of oxytocin twice a day. Normally it should be, sorry, it's not the good one. I'm sorry, I made a mistake. So unfortunately, we will not see. This baby is very active. Only after 7 days of receiving oxytocin, it can respond to the doctor who was examining him, and he tried to move, and he has a really good wheezing. So the question was, does oxytocin modify infant behavior, and can we observe long-term effects? So I'm sorry, I just realized that it's in French, so I made a mistake. That's the reason. So oxytocin improves sucking and swallowing and social engagement in infants, and so we also demonstrate in 2011 that in adults, oxytocin improves trust, anxiety, and disruptive behavior. And the question is that if we treated them early in life, do we have long-term effects? So we don't have the answer, of course, but we have some data after 3 years following these babies who had been treated in 2013, and they are 3 years of age here. The only thing that we observe at this time is that albeit crawling is not observed, or very rarely observed in these infants, when they were treated with oxytocin, 81% of them achieved crawling compared to 13% of the non-treated infants. And now we are data on 4 years old of these children. We have compared it also with untreated child with Prader-Willi syndrome. And we are analyzing the results, but what we observe is we have very better communication still when they were treated early with oxytocin. And just to finish, there is links between oxytocin and ghrelin. I just show you that oxytocin increased the levels of circulating acylated ghrelin in infants. But it's also in the literature that they have comparable effects on behavior, oxytocin, and through the oxytocin receptor and through the ghrelin receptors on mood, social abilities, anxiety, memory, learning, appetite, metabolism, and brain expression pattern. The only thing that we have now, we don't know if ghrelin has social effects on social abilities. We don't know if the ghrelin have effects on learning, but as you can see, there are comparable effects through acting through this kind of receptor. And interestingly, we have also data on the possibility of heterodimer formation between these two receptors, particularly in hypothalamic neurons. And interestingly, when there are formation of heterodimer, the trafficking of the oxytocin receptor and the signalization of this receptor is modified. So now it's very well documented that the formation of heterodimer is a very important thing in controlling behavior, and particularly controlling feeding behavior. Very recently, last year, we also had a very interesting publication showing that the same effect on rescuing high lethality in another mice model, but not using oxytocin and using ghrelin analog, completely recapitulate the effects of oxytocin in infants. So administration of this analog particularly to another model of Prader-Willi syndrome completely rescue a postnatal lethality, which completely demonstrate the links between oxytocin and ghrelin. And of course, I'm not going to this last slide because there are many approaches, different approaches to treat Prader-Willi syndrome from many years now, from 5 or 6 years, targeting different receptors acting on feeding control. The setmelanotide target the MC4R receptor. Cannabinoid also is cannabidiol is a very interesting drug. And the other, carbetocin, is an analog of oxytocin. And diazoxycholine is also a drug that modified the trafficking of the ghrelin receptor. And nivolotide I have just shown you the results. I have to thank my team first and all the collaborators and you for listening to me.